later, yeah, I will post the video, yeah, link online, and you can also, yeah, start it, yeah, after the yeah class, okay. All right, so yeah, for today's kind of training, I will talk about the yeah, HPLC and those the nucleic acid, okay, mainly DA, and take about like maybe 1 hour around, okay. And if you have any question, okay, so you can speak out, okay. So yeah, because yeah, we don't have too many people, okay, and also you can type in the chat window, okay. So yeah, if I saw that, okay, I will answer your question. All right, so first section is kind of yeah HPLC. Okay, so yeah, here's the very simple kind of yeah the scheme of your HPLC setup. Okay, so you can see kind of there's kind of yeah the mobile phase. Okay, and also pump yeah the yeah solvent into yeah, the auto sampler. Here you yeah will do the injection of your sample, and then the yeah. Eluent, okay, the solvent bring your sample pass through yeah HPLC column, and then yeah yeah your sample gets separated under the HPLC column, okay. And afterwards, okay, so yeah there's yeah will be detector yeah connect after yeah the column, okay, so yeah tell yeah which sample come out which, and then eventually you see the all the chromatogram on the yeah computer, okay. So HPLC stands for high performance liquid chromatography. And sometimes also yeah refers to a high pressure liquid chromatography. Okay, so yeah, it's very powerful tool, very powerful tool, okay, very useful. Okay, so later I'll talk about this kind of application. Okay, and here's kind of yeah some components for the HPLC. I just talked about it. Okay, and then I will show you guys one video. Okay, so yeah, see, okay, hopefully it's can play. HPLC, high performance liquid chromatography, but could equally well stand for high pressure liquid chromatography.
It's a little quiet.
It is used for separating. Can you turn your volume a little bit higher? What separator requires can also be used to find the relative amounts of different components in a mixture. HPLC works on the same principle as paper chromatography, here shown speeded up. A liquid, called the mobile phase, moves past a solid, the stationary phase. In paper chromatography, this stationary phase consists of water molecules bound to the cellulose in the paper. The mobile phase carries different components of a mixture, called the sample, along with it at different rates. How fast each one moves depends on its relative affinity for the mobile and stationary phases. For example, if the mobile phase is more polar than the stationary phase, the more polar components of a mixture will tend to move more quickly than the less polar ones. In HPLC, the stationary phase is a solid packed into a column, like one of these. This particular column contains silica particles to which C8 hydrocarbons are attached, making the stationary phase non-polar. In paper chromatography, the solvent moves along the paper by capillary reaction. In HPLC, the liquid is forced through the column by high-pressure pumps. The whole apparatus looks like this. These bottles contain solvents. Two solvents can be mixed in any proportions to give a mixture, the liquid phase, a suitable polarity for the separation that is being done. In this case, one solvent is water, very polar, and the other ethane nitrile, less polar. The operator can decide on a mixture with the correct polarity for the separation she is doing. These are the pumps. They produce a pressure of 15,000 kilopascals, 150 times that of the atmosphere, hence the name high pressure liquid chromatography. If a single sample is to be run, it is injected into the solvent stream here, in the injection port, via a hypodermic syringe.
Alternatively, several samples can be run in succession by loading them into this auto sampler, which will run them in order without any human intervention. Pumps force the mixed solvents through the column. The solvent emerging from the column, carrying the separated components of the mixture, passes into the detector. Here, a beam of ultraviolet light shines through it. This light is set at a wavelength that is absorbed by all the components to be separated. When the detector reading drops, a component that is absorbing UV light is coming out of the column and passing through the detector. Many alternative types of detector are possible. This one measures refractive index. The time that each component takes to come off the column is called its retention time and can be used to help identify it. Here, the HPLC instrument is being used to separate a mixture of two steroids used in a pharmaceutical preparation. The column chosen is packed with a non-polar solid. The tail of the molecules represent hydrocarbon chains C8H17. Having chosen the solvents, detector wavelength, flow rate, a single sample is run by injecting about 20 microliters into the injection port. The more polar component comes off the column first, followed by the less polar. The peak at retention time, 1.5 minutes, represents other ingredients used in formulating the product. This is the pharmaceutical product, and behind it, its chromatogram.
All right. Sorry, okay, so yeah, because it's here not in the class, okay. If it's in the classroom, okay, so maybe you guys can hear it better, okay. And so this video I will post here its link online, okay, so we can watch it later. Okay. All right, okay, so the HPLC actually belongs to the column chromatography, okay, so and it's there's just there's retention and also absorption and distribution okay between kind of stationary phase, okay. And separation of the absorbed acceptance okay, just based on the mobile phase, okay. So if there are kind of weak binding with the stationary phase, okay, and then the mobile phase can bring down the substance quickly, okay. And then afterwards, okay, you just recover the individual components, okay, one by one, okay. And you can also quantify kind of the individual components, okay, just based on the detector. And here's some the basic setup. Okay, so just keep in mind, okay, so separation is kind of depends on distribution. Okay, so ability between kind of stationary phase and also mobile phase. Okay, so stationary phase is just the solid part in the HPLC column. Okay, so actually, most common one nowadays for the HPLC the packing materials is kind of C18K, C18K. C18 is kind of C18 kind of carbon chain they packed on linked on the silicon gel, okay, silicon K. And also there are kind of C8 also kind of just regular silicon, okay. And mobile phase, okay, so normally there's the one aqueous phase, one's kind of organic phase, okay. And normally the organic phase has kind of more kind of washing power or kind of distribution power than the aqueous phase, okay. Are have to kind of release the compound bind on the station phase, okay. And here's kind of maybe small cartoon, okay, show kind of separation of individual components along the HPLC column, and eventually they pass through the detector one by one, and then we can see kind of the chromatogram kind of peaks one by one. All right, okay, here comes kind of most two important information given by HPLC. So one is the retention time, okay, so also called TR, okay. So once the HPLC condition kind of yeah set okay so yeah for example yeah column okay flow rate okay so yeah gradient and also the mobile phase whatever okay and for the yeah, same sample okay same kind of the component okay so they should have kind of same return time okay so that's help to us kind of identify individual components okay so for example yeah for this unit okay so yeah we first need to identify the yeah return time for ATCG, okay, so yeah, different kind of nucleoside, okay. And then once we figure out, okay, so yeah, if you, next time, yeah, if you have some unknown sample after digestion, okay, so yeah, yeah, you kind of yeah, at the same position, you show the same peak, okay, so that you can identify yeah, what's yeah, nucleotide, okay, so yeah, nucleoside, okay. And yeah, another important information is the yeah, area. Okay, so area, or sometimes we'll call it kind of integration. Okay, so yeah, the peak area as kind of yeah, yeah, can be used to yeah, quantify yeah, the individual components. Okay, and but yeah, for individual components, okay, they have different ability to absorb yeah, UV light. Okay, so normally we use yeah, UV detector for yeah, DNA digestion. Okay, so yeah. And so that's come kind of yeah beer lambda law. Okay, so let me do this. Okay. Alright. Can you guys see my document camera? Yes. Okay.
Yes.
All right. Thank you. Yeah. So beer lambda law is kind of here yeah that's here the absorption. Okay. So here yeah equals kind of here lambda times kind of here the C times kind of L. Okay. So L is kind of here cell length. Okay. So yeah. So this one won't change. Okay. And then next kind of yeah concentration. Okay. So that here will change. Okay. And the next one kind of here this is here yeah epsilon k that here yeah we call kind of yeah extinguishing coefficient case. And so all kind of here yeah nowadays people call kind of here yeah absorption yeah yeah cofactor or whatever. Okay, so yeah. So yeah, different compound will have a different this yeah number. Okay. And so yeah, for example, yeah, for our yeah, the DNA digestion project, okay, so yeah, ATCG, okay, so yeah, the all four different nucleoside will have different kind of yeah, yeah, epsilon, okay, so yeah, extinguishing coefficient, okay. And so your work is kind of yeah, try to figure out yeah, individual kind of yeah, yeah, nucleoside kind of yeah, yeah, extinguishing coefficient, okay, so yeah, here in our project we call it kind of cofactor, okay cofactor. Okay, so that's here yeah. Please keep in mind. Okay, so yeah, that's yeah, actually kind of yeah, pretty important. All right, let's go back. Okay, and here's kind of here yeah, one example of here this one example of here yeah, UV chromatogram. Okay, and yeah, UV kind of yeah, yeah spectroscopy. Okay, and for yeah chlorophyll. Okay, so yeah, and it has kind of yeah two peaks. Okay, so yeah, yeah that's we can label as kind of lambda max. Okay, and then for individual this peak they have its own kind of yeah yeah epsilon max. Okay, so yeah that's here yeah extinguishing coefficient. Okay, so yeah the molar coefficient. Okay. And so yeah, for our kind of yeah DNA project case, yeah, we use it kind of yeah, yeah 260 nanometer to monitor all four yeah nucleoside. Okay. And so yeah, like I said, the most important kind of yeah, you need to figure out kind of yeah, yeah cofactors. Okay, so yeah, for individual yeah, nucleoside. Right. And HPLC kind of very useful, okay, very powerful tool. Okay, so yeah, as long as I know, okay, at least maybe 4 or 5 kind of my friends or my colleague, okay, so yeah, they know how to run HPLC, they find jobs, okay, so yeah, it's yeah. So if you are a kind of HPLC expert, okay, so it's very easy for you find kind of yeah yeah academic job, okay, or kind of yeah the industrial job, okay, so yeah, it's a very powerful tool, okay. And it has many applications. Okay, here are just kind of yeah examples. Okay, for example, kind of yeah, pharmaceutical. Okay, so yeah, for example, yeah, yeah, identify some kind of yeah counterfeit kind of drugs. Okay, so yeah, the product. Okay, yeah, this morning I just gave one example to my yeah organic chemistry yeah yeah lecture students. Okay, so yeah, for example, okay, so some yeah pharmacy yeah kind of purchase some the medicine from kind of India. Okay, so yeah, but yeah, there are kind of yeah inactive component in the yeah medicine. Okay, and then that's easy to identify. Okay, you just kind of run HPLC for that kind of medicine drug with kind of standard kind of medicine drug. Okay, so yeah, to track their kind of HPLC trace and also integration. Either identify there are kind of some actual components. Okay, or some kind of last kind of active kind of components. Okay. And environment case, so for example, some kind of textile kind of industrial case, so dump the wastewater in the in the lake or kind of in the river, okay, and then you just run HPLC, or you can easily identify others kind of the dyes in the river water, okay, so in the wastewater, okay. Forensics, okay, so for example, that you can use the people use HPLC identify the illegal drugs in some kind of people, okay, so just by analyze their kind of urine or kind of urine or kind of sweat, even some kind of it's just hair, okay, easily I figure it out, okay, so or maybe some kind of athletic people, okay, so use anabolic kind of steroids, okay, to improve their performance, okay, so that's just pretty easy check their kind of the urea, okay, so by HPLC can easily identify what's the illegal component, okay. And last example kind of food and also flavor, okay, so that's normally for the kind of quality control, okay, so you analyze kind of sugar components in the juice, okay, so and then how much it is, okay, so pretty easy, okay. And like I said, okay, so in this units, okay, so one important thing is kind of we will learn the both theory and also the practical application of the HPLC. Okay, I will teach you kind of some kind of really hand-on kind of experience for the HPLC. Okay, so very useful. Okay. The bottom are kind of two figures. Okay, so last is kind of HPLC forest okay, in the pharmaceutical company. Okay, so quite often in the pharmaceutical company, even you cannot see kind of other analytical instrument. Okay, so like an animal. Okay, but definitely they should have kind of pharmaceutical kind of definitely should have kind of HPLC instrument. Okay, so so that's I have been one pharmaceutical company. Okay, the whole floor. Okay, they have maybe like a 100 HPLC. Okay, so that's take whole floor. Okay, so that's a lot. Okay, and then also for our HPLC kind of quite small, okay, but industrial, okay, so they use kind of HPLC to isolate kind of compound in kilogram scale, okay, so that's on the right you see kind of the industrial kind of scale, HPLC, okay, so that's the column kind of longer than people, okay, so that's pretty impressive, okay, so. All right, okay, so next, we move to some basic knowledge about nucleic acid and those mainly about kind of DA, okay. Right, so DA is kind of stand for deoxyribose nucleic acid, okay. And so that's one types of the nucleic acid. We'll talk about now other types of nucleic acid, okay. So and the basic units of DNA is called nucleotides, okay. So nucleotides has kind of three components, okay. Phosphate and nuclear base. And nuclear base kind of two kind of nuclear base. One kind of based on the purine, one based kind of the pyrimidine, okay. And another one kind of the third component is just sugars, okay. So right, we do one by one, okay. So first one is the nuclear base, okay. So there are three nuclear base based on the pyrimidine, okay. So one kind of uracil, second one kind of thymine, and also the last one kind of cytosine, okay. So just please keep in mind, okay, uracil only exists in RNA, and then thymine only exists kind of in DNA, okay. And then two nuclear bases are based on purine, okay, so one's kind of adenine, okay, another one kind of guanine, okay. All right, and then next kind of the sugar, okay. So you can see kind of normally people use the numbers to label the relative position in the nuclear base, okay, 1, 2, 3, 4, okay, and then for the pyridine case, purine case, so nuclear base kind of up to kind of 9, okay. So to distinguish those number on nuclear base, okay, people use kind of number prime, okay, so to label the sugar, okay. So that's actually pretty important, okay. So stop the sharing, and then we need to show you guys on the okay. So whatever I write on the kind of the paper, please also record it on your notebook, okay. And then the slides you don't need to record, okay? The slide I will upload online, okay? Yeah. Right. So for the sugar, okay, so yeah, also called kind of yeah the yeah pentanose, okay? So yeah. Right. So one difference yeah between the DNA and those R is kind of yeah the yeah two prime position, okay? So yeah, or yeah, like I said, kind of we use yeah the yeah number prime, okay, labeled position, okay? So yeah, here this yeah first position, okay? So normally yeah we connect with yeah nuclear base, okay? So yeah, that's yeah this one's gone, okay? Become base, okay? Nuclear base, okay? And then that's where we label as one prime, okay? And then we go to kind of here the yeah clockwise, that's yeah two prime, three prime, four prime, and five prime. Okay, so that's here the totally five carbons. And the one difference between kind of here the yeah DNR is kind of here for the sugar of your DNA. Okay, the two prime is kind of here the yeah just hydrogen. Okay, no OH group. Okay, so that's why DNA called kind of here deoxyl kind of ribose. Okay, so yeah because there's kind of here no OH group. Okay, so yeah. Right, and then yeah, please record it. Okay, so yeah, because yeah, later we need to yeah, explain kind of yeah the more thing based on yeah the sugar yeah the relative positions. Right, okay, okay. So here's kind of yeah the difference between kind of yeah the yeah DNA and also RNA kind of yeah ribose. Right, and still hide this one. So once the yeah sugar connect with the yeah nuclear base, okay, we generate kind of yeah nucleoside, okay, nucleoside, okay. And so yeah, normally the sugar connect the yeah the yeah nuclear base yeah through yeah we call kind of yeah beta glycosidic kind of bond. Okay, so that's this bond okay between the yeah nuclear base nitrogen and also yeah one prime carbon. Okay. So why it's called beta? Okay, because yeah, this bond kind of yeah center. Okay, so yeah, the yeah, if it's going up, okay, we call it beta. Okay, and if it's going down, okay, so this bond going down, that's we call it kind of alpha. Okay, so for all the natural kind of yeah, the DNA also RNA nucleic acid. Okay, so yeah, all a beta yeah, the yeah, kind of yeah, glycidic kind of yeah bond. Okay, so yeah, that's yeah, please keep in mind, okay, all going up. Right, and then yeah, yeah, we can yeah, assemble kind of yeah nucleotides. Okay, so nucleotides kind of yeah, yeah, we add yeah phosphate. Okay, either on the yeah five prime or kind of three prime. Okay, so yeah, yeah, bottom one kind of yeah the phosphate kind of on the five prime. Okay, and so in this case, actually, kind of we can name this compound as yeah, the five prime mono yeah phosphate adenosine kind of five prime monophosphate. Okay, so five prime indicates kind of this phosphate add on the yeah five prime carbon. Okay, certainly you can also add kind of yeah phosphate on the three prime. Okay, so fill out yeah the bottom one. Okay, so that's yeah uridine three prime monophosphate. Okay. And so in addition to yeah monophosphate, okay, you can add kind of yeah the yeah two phosphate and then triphosphate, okay, so yeah three phosphate, okay, on the yeah the yeah nuclear side, okay, so that's yeah kind of yeah the yeah NDP, okay, NTP, okay, N indicate kind of yeah the yeah nuclear base, okay, so yeah depends on the individual, okay. So one of the yeah, most famous kind of yeah, the yeah, nucleotides kind of yeah, the ATP. Okay, so yeah, adenosine kind of yeah triphosphate. Okay, so yeah, there are kind of three phosphate okay, attached on the three prime of yeah, the yeah adenosine. Okay, so that's yeah, energy transfer yeah the chemicals. Okay. Right, and here's kind of yeah, the little bit summary of yeah, nuclear base, nucleotides, and also yeah, nucleosides. Okay, just keep in mind, okay, so yeah, nucleosides, okay, plus phosphate, okay, we generate kind of yeah, nucleotides. Okay, so nucleotides is yeah, the yeah, basic unit for the yeah, DNA. Okay, basic unit okay, for the DNA. Right, okay, so once yeah, we have yeah, the nucleotides, okay, how do we assemble yeah, the nucleotide together into yeah, the DNA strand? Okay, so that's yeah, we talk about kind of yeah, primary structure of DNA. Okay. And for primary structure of DNA case, yeah, pretty much yeah, the nucleotide case, so the base and sugar connect each other through kind of yeah, the phosphate at yeah, three prime and also five prime. Okay, so yeah, what's that mean? Okay, so yeah, yeah, phosphate okay, one end connect yeah, three prime of yeah, the one yeah, nucleoside and connect yeah, five prime of another nucleoside. Okay, and then one by one, okay, eventually you generate kind of yeah, the yeah, DNA strand. Okay. And so yeah, just yeah, please one thing, please keep in mind, okay, because there are kind of three prime and five, three prime and also five prime, okay. So then the DNA really has sequence, okay, sequence, okay. So you cannot reverse order, cannot reverse order, okay. So traditionally, okay, so we talk about kind of yeah, DNA sequence. That's actually start from yeah, five prime, okay, five prime end, and then we go down, okay, three prime end until end, okay. So yeah, that yeah, yeah, for example, yeah, this is a very short DNA case. So yeah, yeah, CATJ case. So CATJ case. So that's the indicate kind of yeah, the five prime. It's kind of yeah, C, and then ended at kind of yeah, G at yeah, three prime. Okay. So that's yeah, please keep in mind. Quite often, yeah, people even ignore kind of five prime. Okay. So yeah, just kind of CATJ. Okay. You have to keep in mind. Okay. They strand start yeah from yeah, the five prime. Okay. So that's yeah, yeah, common mistake yeah for some kind of yeah, yeah, beginner. Okay. So yeah, when they order kind of strand, okay, so yeah, they forgot kind of yeah, it's either three prime or five prime, okay, so yeah, that's happened kind of yeah, when I was yeah, grad student, okay, so yeah, one student in my group, okay, so yeah, the boss asked him kind of yeah to order some DA strand, okay, so yeah, the order kind of totally reverse the strand, okay, so yeah, forgot kind of yeah from five prime to three prime, okay, so that's yeah, please keep in mind, okay, all the DA we yeah top of the sequence always start from five prime, always start from five prime to three prime. Okay. Right. Okay. So yeah, but for the natural DNA case, so that the not in the single strand, not kind of yeah the straight line case, so yeah, it's in the yeah. Most time kind of in the double helix case. So double helix belong to kind of yeah, the secondary structure of yeah, the DNA. Okay. So this yeah, yeah, double helix kind of yeah, the two DNA strand. Okay. So yeah, if there are yeah, the base can match each other. Okay. So A match with T and also yeah, the C match with A, C match with G. Okay, and then they can assemble into kind of here right-handed kind of here the double helix. Okay, so this here the yeah, double helix model kind of here the yeah, proposed by yeah, Watson and Crick. Okay, so that's one of your most fundamental kind of yeah, breakthrough in the yeah, last century. Okay, and so yeah, like I said, okay, so yeah, yeah, A and T can match together, okay, through kind of yeah, yeah, form kind of yeah, yeah, hydrogen bonding, okay, form kind of base pairs through hydrogen bonding, okay, and then A and T each contribute one hydrogen bond donor and one hydrogen bond acceptor, okay, and then can assemble into yeah, can form a two hydrogen bond, okay. And for yeah, C and G case, yeah, yeah, G can yeah, contribute yeah, two hydrogen bonder donor and then one hydrogen bond acceptor, and then C contribute kind of yeah, two yeah, acceptor, one donor, okay, so can form kind of yeah, three hydrogen bond, okay, and clearly kind of yeah, yeah, C and G can form kind of yeah, the more hydrogen bond. That means kind of yeah, they are bonding kind of stronger than A and T, okay, so that's the answer is yes, okay. And then yeah, yeah, binding affinity between A T base pair kind of yeah 10 kcal per mole, okay, and but yeah CG kind of much higher, okay, so yeah 17 kcal per mole, okay, yeah. All right, and then once yeah two strand, okay, so yeah they're all the base yeah match each other, means kind of A match T and also yeah C match G, okay, then they can form kind of yeah secondary structure double helix, okay. And just one thing, please keep in mind, okay, here the double helix condition are anti-parallel, anti-parallel. Okay, so one strand kind of yeah from yeah five prime to yeah three prime, and then another strand kind of yeah yeah it's opposite directions. Okay, so yeah that's here yeah something please keep in mind. Okay, so maybe in the yeah quiz. Okay, so on yeah Thursday I will have one question ask kind of yeah draw the yeah complementary strand for this yeah DA sequence. Okay, so you have to keep in mind, okay, so it's a complementary kind of yeah, the anti-parallel, okay. It's kind of yeah, reverse sequence, okay. So yeah, that's yeah, please keep in mind, okay. All right, okay, so yeah, there are some models to show yeah, DNA duplex, okay. So yeah, the easiest ones kind of yeah, the on-road kind of just yeah, this kind of yeah, planar structure, okay. Clearly show kind of yeah, the yeah, DNA base pair, okay. But yeah, the more reality one kind of yeah, on the right, okay. So that's yeah, the yeah, kind of yeah, showing really exactly kind of yeah, DNA double helix, okay. All right. And here I will stop like maybe half minutes, let you guys record some kind of important yeah, yeah, numbers, okay? The first number is kind of yeah, the diameter of yeah, yeah, here's kind of yeah, BDA, later I'll talk about kind of ADA, okay? So yeah, diameter of yeah, yeah, DNA is about yeah, 20 angstrom, okay? Also 2 nanometer, okay? And then yeah, the base distance okay, between kind of the distance between kind of two base pair about kind of the 3.4 Armstrong. Okay. And because for each one full turn of your DNA is about 10 base pair, okay, 10 base pair, okay. So that means kind of one full turn of your DNA is about the 34 Armstrong, okay. Yeah, please recall these three numbers, okay. So so I made testing kind of in the quiz on Thursday, okay. Yeah. All right, and for the BDA, clearly you can see there are kind of two growths. Okay, so one kind of little bit bigger, we call it kind of major growth. Okay, another one kind of little bit narrow, we call it kind of minor growth. Okay, so that's also pretty important. Okay, sometimes kind of if we enzyme bind with DNA, okay, so normally through kind of major growth. Okay, because that's kind of bind deeper in the DNA. Okay. All right, okay. So next kind of another model for DNA. So this one actually kind of more close to real structure of DNA. This one called kind of field space kind of model. Okay, and clearly you can also see kind of major growth, kind of minor growth case. All right, and so for the DNA duplex. Okay, so so far we just only talk about kind of the BDNA. Okay. BDNA is kind of the most common DNA, the secondary structure. Okay, so normally existing kind of dilute aqueous solution, and the right-handed kind of helix. Okay, so those data are already talked about it. Okay, so and so there are also two other main kind of DNA duplex form. Okay, so one we call them ADA. Okay, ADA also kind of right-handed kind of helix. Okay, so but much thicker and also much shorter than the BDNA. Okay, so it's still kind of 10 base kind of for one full turn. Okay, so and the third one kind of called ZDA. Okay. ZDA is kind of left-handed kind of duplex. Okay, so that's very uncommon, very uncommon. Okay, so only very special conditions. Okay, so you use kind of the special kind of salt condition. Okay, you can generate kind of ZDA. Okay. And here's kind of the figure for the ABZDA. And so you can see clearly the BDA is kind of the longer and also smaller than kind of ADA. Okay. And then you can also see clearly ZDA is kind of the right-handed case. Yeah. And the bottom three figures are just the top view of the three kinds of DNA. Right. Okay. So but for most time, okay, for most time, kind of the DNA won't exist at kind of just the long duplex string. Okay. So so for example, for all kind of the human kind of DNA. Okay. So if it's kind of existing kind of long duplex string. Okay, that's about kind of 6 feet. Okay, that's impossible kind of fit in our kind of cell. Okay. So reality, most time DNA will stay at kind of tertiary structure as highly packed kind of structure, okay, for DNA. And so normally kind of refers as kind of supercoil, okay, supercoil, okay. So there are different kinds of supercoil, okay. So this one kind of circular DNA, okay, it's just kind of the 3' and also DNA duplex kind of kind of connect to each other, form kind of circle, okay. And but in our cell, okay, how come kind of the 6 feet kind of DNA duplex can pack in kind of the very tiny cell, okay? So that's actually form kind of the chromatin, okay, chromatin, okay. So chromatin is kind of the DNA duplex, okay. So circulate around the protein called kind of histone, okay. And then in a kind of bead-like structure, and then further packed down, okay. That's generally kind of chromatin, okay, chromatin, okay. All right, here's gonna figure some the circular DNA and some supercoil. Okay, on the bottom is kind of DNA duplex surround the histone generate the chromatin. Okay, actually chromatin can further pack down. Okay, generate kind of the chromosome. Okay, chromosome. Okay, and I should have a very nice video. Okay, hopefully I can play it.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. The nucleosomes are packaged into a thread known as chromatin. This fiber is then looped and further packaged using other proteins which are not shown here. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope: chromosomes. Chromosomes are not always present. They form around the time cells divide when the two copies of the cell's DNA need to be separated. At other times, as we can see now after the cell has divided, our DNA is less highly organized. It is still wrapped up around the histones, but not coiled into chromosomes.
Again, okay, so yeah, if you cannot hear this video, okay, so yeah, you can download the video kind of later, okay, watch it, okay. Right, so next is the RNA, okay, so RNA is kind of similar structure, okay, so yeah, to the DNA, okay, but there are kind of three differences, okay, so yeah. First one is kind of the sugar unit of the RNA is actually kind of ribose, okay. So ribose means kind of 2' should be OH group, okay? 2' should be OH group, okay. And the different kind of hydrogen on the kind of 2' of the DNA, okay. And the second difference kind of the pyrimidine kind of base of RNA is kind of reversal, okay, and then and but yeah, for the DNA, it's kind of the thymine, okay. So that's we just talk about it, okay. And the third difference kind of DNA normally kind of in the duplex form, okay, but RNA actually kind of normally in kind of single strand form, okay. Yeah, those three, please keep in mind. I made testing kind of the future quiz, okay. So, yeah. Right, okay, so yeah, for the RNA actually has kind of more function than DNA. DNA normally is kind of for the genetic information storage, okay. So RNA kind of depends on these rules kind of have different function, okay. So here are kind of three examples, okay. The first one kind of rRNA, okay. So that's actually kind of one important component for the ribosome kind of some small kind of the functional kind of component, okay, can use as kind of the protein synthesis, okay. And so yeah, second type kind of RNA is the tRNA, okay. So that's this tRNA are coded to special kind of nucleoside, okay. Sorry, coded to special kind of the amino acid, okay. So then they can bring kind of this special amino acid kind of to ribosome, okay, and then make proteins, okay. And the last one kind of mRNA. Okay. mRNA is just normally use DNA as template, okay, make mRNA, okay. And then in the ribosome, okay, use mRNA as template to make proteins. Okay. And the gene and also the DNA code, okay. So DNA kind of just section of the DNA coded for certain proteins, okay. And then each unique the gene kind of has a unique sequence of the DNA base, okay. And so that means kind of unique sequence of the DNA base, okay. So coded for certain kind of types of proteins, okay. And eventually this the combination of proteins give us kind of unique prototype, okay. So for example different color, okay, different kind of the hair, okay, so different skin, whatever, okay. So and so if we use the DNA as template kind of make the mRNA, okay, this process we call kind of transcription. Here's one thing, please keep in mind, condo, okay. Condo means kind of three DNA base pair coded for one new amino acid, okay. So that's we call kind of this triplet as the condo, okay, condo. And here's some very basic kind of genetic code, okay. So for the different combination of nuclear base kind of three nuclear base, okay, you coded for different kind of amino acid, okay. And then we can a little bit of summary about kind of central dogma, okay. So pretty much the DNA can replicate itself, okay. So we make another DNA, okay. And then DNA store all the genetic information, okay. And then use DNA as template we can make here the mRNA, okay, and then just kind of cell, okay, so the mRNA as the template, we can use that kind of to make here the protein. This process we call kind of translation, okay, so from DNA to RNA we call kind of transcription, okay, and then RNA to protein we call kind of translation. All right, okay, so next important components is the enzyme, okay, so for my units, okay, we use two enzyme, okay, so here I will talk about kind of three kind actually kind of only two main kinds of enzyme, okay, so but I all list kind of other two kinds, okay, so all totally four, okay, so first one kind of DNA polymerases, okay. So DNA polymerase can just kind of assemble the DNA nucleotide together, make kind of DNA kind of string. Okay. And the second one kind of kinase and also the alkaline phosphatase. Okay. So this just play around kind of phosphate part of the nucleotide. Okay. And then third one kind of nucleus. Okay. Nucleus kind of cleave out kind of DNA. Okay. Break up DNA kind of into nucleotides. Okay. So last one kind of topoisomerase, okay, that's kind of packing of DNA. Okay. So we mainly focus kind of okay, actually the number is wrong. Okay, so the alkaline phosphatase and just kind of second and also the third type. Okay, so the two enzyme we use in our project, one is kind of based on the alkaline phosphatase, okay, another one kind of nuclease. Okay. And for the enzyme to work, okay, so you have to use buffer, okay, so that's pretty important for the enzyme activity, okay. And so for buffer, okay, so there are some further falling kind of the conditions you have to kind of make sure kind of when you prepare the buffer, okay. So first kind of this buffer, kind of the pK about kind of 6 to 8, okay, so that's close to our kind of the body bio environment, okay. And the second had to be kind of chemical invert, okay, so you don't want some kind of chemical reaction happen kind of when you add this buffer in, okay. And third had to be polar, okay, so that's just to make sure kind of soluble to the old enzyme and also not membrane permeable, okay. So next one just not toxic, okay, and then not expensive, okay. And the last one kind of salt, okay, so and also temperature indifference, okay, so you don't want kind of change temperature and then dramatic kind of change buffer kind of the pH value, okay, and also no influence kind of by salt, okay. And for our kind of DNA digestion project case, we will use kind of the trace buffer, okay. The trace pK is kind of 8.0, okay. And then the trace full name called kind of trace the hydroxymethyl amino methane, okay. So this one, let me see, okay. So probably I need to show you structure, okay, because that's pretty important the component case right, trace, remember the name called kind of trace hydroxyl methyl amino methane, okay. So if you took my organic chemistry, you should know, okay. So when we draw the structure based on name, okay, we do the backward, okay. So first, we draw the methane, okay. So methane should attach the full hydrogen, okay. But you don't touch it now, okay, because there are some other thing you will touch, okay. Amino methane, okay, that means there's kind of amine group on it, okay. And next kind of the trace hydroxy methyl, okay. Hydroxy methyl is means there's kind of methyl group, okay. These three, okay, but there's the hydroxy under the methyl, okay. So that's the hydroxy methyl, okay. Trace means there are kind of totally three units, okay. Three units on this the central carbon, okay. So that's the trace, okay, trace. Okay, so this one should be CH. Carbon. This one should be carbon. Okay. And but quite often you should see kind of trace pK is kind of 8. Okay, so but quite often you should see kind of trace HCl. Okay, so reality is kind of form of salt with the hydrochloric acid. Okay, so when you add kind of HCl in the trace. Okay, so then we just kind of ammonium salt and then followed by kind of counter ion, okay, Cl, okay. So that's the trace HCl, okay. So this is a real buffer. This is real buffer, okay. Yeah. make sure you record the structure. Okay. All right. Hope you guys get it. Okay. Right. Trace HCl kind of the buffer is PK about kind of 7 to 8. Okay. Right, and then also some kind of the other component for the buffer. Okay, so if you want to make it enzyme active. Okay, so first kind of buffer, and then are kind of also some salt. Okay, so that's just balance the chart. Okay, so because for the DA and also protein are some the amino acid or kind of nucleic acid. Okay, so you need balance chart. Okay. And more important, some kind of divalent the metal ion, okay, so like magnesium, okay, so that help to activate the enzyme. So for example, in our kind of DNA digestion project, okay, so we will use the magnesium chloride. And then glycycle, okay, so glycerol, okay, glycerol actually kind of for storage, okay, so stabilize the protein structure, okay. And the EDTA okay, sometimes kind of just help to kind of chelate the divalent cation, okay, so it's also helpful kind of storage, okay. And so next kind of some sulfur compound, okay, so for example the diethyl three okay, so those just help kind of reduce some sulfur kind of side chain on the protein, okay. And then there are some other last important component like most rescued proteins, okay, like bone serine kind of albumin, okay, and also some kind of ATP and also the AADH, okay. All right. All right, so here we'll talk about kind of nucleus first, okay? So nucleus, okay, so there are two types of nucleus, okay? One is we call the axial nucleus, okay? So this one kind of cut the DNA strand from end of nuclear DNA strand, okay? End of the DNA strand, maybe from prime or for kind of the three prime, okay? So that's why I call it axial, okay? Another type of nucleus called kind of endonucleus, okay? So this one will cut kind of the DNA from middle, okay? So just the break up in the middle of the phosphodiester bond, okay? So one type of kind of most famous kind of endonuclease kind of restriction enzyme, okay? So that's we'll talk about later, okay? And here I give you two examples of axial nucleus, okay? So one is three, okay, three example, okay. One is kind of axial nucleus three, okay. So this one can only work on the double strand DNA, okay. So cut from the 3 prime to 5 prime. Another one kind of the axial nucleus, okay. So that's one only work on a single strand DNA, okay, cut from kind of 3 prime to 5 prime. And then that's useful kind of just the more the on extended kind of the primers kind of in the DNA strand. Okay. Right, and then third types of is actually the endonuclease. Okay, so endonuclease. Okay, I give you two examples. Okay, one is Dynase one. Okay, Dynase one. Okay, so Dynase one is just leave double strand DNA randomly. Okay. Leave the double strand kind of randomly, okay, and depends what kind of the salt, okay. So if you use the manganese, okay, that's you cleave for kind of the double strand, okay. But if you magnesium, okay, that's only cleave kind of the single strand, okay. So very useful for define the kind of binding site for the certain enzyme to DNA, okay. So here I give you one example, okay. So for example, this is the double strand DNA. Okay, so I label one strand with the P32, right? And then if I apply kind of Dynase one, okay, so that's cleave the DNA at any positions, okay, any position, okay. And then you can generate kind of different lengths of the DNA strand. And so then we just run kind of chromatography, kind of in the gel electrophoresis, okay. So you can see kind of the depends kind of different the strand lengths, okay. I can clearly identify kind of the base, okay. So the sequence, okay, under the DNA sequence, okay. And so if there's the one enzyme bind with this DNA double helix, okay. And because this will hinder kind of cleavage of the Dynase one, okay, then it will bypass the binding part, okay. And then if you apply the Dynamics one, okay, so then that's some kind of part will blank, okay. So and because block the cleavage of Dynamics one, okay. So afterwards if you draw kind of electrophoresis, okay, you can clearly see kind of what's binding site for this the enzyme, okay. So that's pretty cool, okay. And the restriction enzyme, okay, so actually also very useful, okay. So that can recognize a certain DNA sequence, okay, only cuts through kind of certain DNA kind of the DNA sequence, okay. Could be kind of kind of the end-to-end, okay, or kind of the mismatch, okay. So that's I use one kind of BAM H1, okay, so very powerful, okay, highly specific, okay. All right, and then for this project case, we will use the first kind of enzyme actually belongs kind of nucleus, okay, more specific called kind of phosphor diesterase, okay. So this one's actually kind of exonuclease, okay. I remember cut from kind of DNA from the 3' to 5', okay. And reality is the enzyme name called kind of the phosphor diesterase 1, okay. And it's extracted from the steak venom, okay. So more powerful, okay. So once you add it in, okay, just click for kind of the whole DNA sequence into the individual nuclear tide, okay. Kind of phosphate still here, okay. But the nucleotide is still two polar, okay, okay, so not easily separated by HPLC, okay. So then we have to apply another the enzyme, okay, cleave for kind of phosphate, okay, and convert the nucleotide into a nucleoside, okay. So nucleoside, okay, and this enzyme we use kind of called a bacterial alkaline phosphatase, okay, BAP, okay. So this actually belongs kind of kinase, okay, type, okay, and only cleave for kind of phosphate, okay, phosphate. And then once we get here nucleoside, okay, and then we can analyze the component by the HPLC. And here's kind of one example. Okay, so we have this DNA sequence. Okay, so we apply kind of two DNA enzyme. Okay, and cleave into individual the nucleosides. Okay, and then we run HPLC. We should see kind of four peaks. Okay, because there are kind of four nucleic base. Okay, so in this sequence. Okay, and then based on retention time, we can clearly figure out what's the kind of which space kind of for this which peak, okay, so in the chromatogram, okay, and then based on kind of integration of individual peaks, okay, and we can figure out kind of how many kind of individual nuclear base, okay, so that's what we'll do in this project, okay. And I when I was graduate, I did a lot of this kind of work. Yeah, pretty powerful, Okay, so here's one project I did before. Okay, so for the normal DNA, okay, so if I apply this two enzyme will cleave into kind of individual nucleoside. Okay, so and then later I make here another kind of the artificial DNA. Okay, so this time case the two preparation, okay, linked by a kind of nylon linker, okay, and then like letters, okay, so double kind of backbone, okay, and then if I apply this the enzyme, okay, that's because although the enzyme can cleave for kind of phosphor diester kind of bond, okay, but cannot cleave for kind of this nylon linker, so that this nylon linker kind of can be still link this kind of the nuclear site, and then that can be detected by the HPLC. Okay, so here are some kind of just the example. Okay, so after digestion, I think see kind of those oligomer of still connected by kind of the 2' nylon bond. Okay, so that's another example. Okay. All right, and then for this project we just handle kind of natural DNA. Okay, so that we supposedly see only 4 peaks. Okay, so this HPLC program can run by another student. Okay, so pretty nice. Okay, you can clearly see kind of the 4 nice peak ATCG. Okay, but which is A, which T, which C, which G. Okay, you need to figure out during the project. Okay, so and then that's based on kind of return time. Okay, and then you can also quantify each individual nuclear base. Okay, based on the return area. Also we could kind of the integration. All right. So actually Dr. Pratt already done its kind of training. Okay, so All right. Okay, so that's we done our kind of lecture. Okay, any questions? No? Okay. All right, so some kind of announcement, okay? And on Thursday, okay, okay, so yeah, we still meet in Harden 302, okay? Harden Hall 302. That's library, okay? And then at 4 p.m. on Thursday. So we'll have yeah unit quiz first, okay? So yeah, for both yeah my units and also Dr. Press units, okay? And then my unit students, okay, please stay, okay, please stay in the same room, okay. So yeah, we'll have kind of yeah further unit training, okay. So yeah, also please yeah bring your notebook, okay. So yeah, I will give some instruction about how do you prepare proposal and how do you prepare kind of lab notebook, okay, and also some kind of other basic training, okay. Yeah, just Thursday, okay, don't forget, okay. And any other further questions? No. All right, if there's no further questions, that's it for the class today, okay? And have good days, guys. Bye.